I think we should go ahead and get started. It's already 5:35, and if everybody could take their seats. I'm Ivor Royston, and I'm the managing partner at Forward Ventures here in San Diego, and also a board member of the Life Sciences Foundation, which is presenting this program today in partnership with Biocom. It's my great pleasure to welcome you to a very exciting discussion here today about the history of genomic sequencing and where we're heading with the new technologies and instrumentation that will allow us in the very near future a whole genome sequencing of every patient where this information can help direct their personalized therapy. And as an oncologist, it's clear to me that as the price point gets down to around $500, which Jay Flatley assures me it will within the next few years, every cancer patient will have his or her tumor sent for whole genome sequencing and compared to the germline for invaluable information that can help direct therapy. Now, for this discussion to take place, one does not have to go very far from here, from home in San Diego, to find the pioneers and the latest technologies and instrumentation that are required for doing gene sequencing, as this is the home here for the Jay Craig Venter Institute, Illumina, and Life Technologies. These three organizations have dominated the gene sequencing landscape in the world today, right here in San Diego. And the leaders of these organizations are also here with us today, and they are our panelists, which will make for a really great event. And my friend and colleague, David Hale, one of the pioneers of the whole San Diego biotech scene, will moderate this illustrious panel. Now, I want to acknowledge our sponsors for today who've provided support. We're very grateful for them. That's Ernst & Young, Biomed Realty, and AIS. And of course, LSF is delighted to co-present this event with Biocom. And Joe, on behalf of the Life Science Foundation, the LSF wants you to know that we look forward to doing more events like this with you in the future. Heather Erickson is the president and CEO of the Life Sciences Foundation. She's about to have a baby, and so she could not be with us today. So in her absence, I just want to take a moment to do what she would normally do, which is to introduce to you the LSF, the Life Sciences Foundation. LSF is a nonprofit organization dedicated to capturing the history, preserving the heritage, and sharing the stories of biotechnology. By collecting and sharing insights from the founding and development of our industry, the LSF seeks to educate and inspire future innovators, to engage the general public, and to provide lay audiences with a robust understanding of the life sciences field. The biotech industry was formed about 40 years ago, so there is an urgency to our mission. We only have a limited opportunity to record these important lessons from the pioneers themselves, witnesses to what I believe will be the 21st century's defining industry. And that is why I chose to become involved with the LSF. And on the back of the program, you'll find more information about our current projects. And if you have not done so already, you can pick up a copy of the LSF magazine, which is located next to the registration desk downstairs. So thank you again for being here this afternoon. It's going to be a great event. And I want to introduce Joe Panetta, President and CEO of Biocom, to say a few words.
Ivor, thanks a lot. I can't begin to tell you how important this organization, Life Science Foundation, is to us at Biocom. This life science cluster here in San Diego has a long, rich, deep history and one that needs to be told more on the world stage. So it's a pleasure to partner with the Life Science Foundation and to be a part of this event. So our mission at Biocom is to accelerate the success of this life science industry in Southern California on the world stage. And we've worked with all the panelists here tonight in leadership in our organization, support as members of our organization, and we appreciate that. One of the things that I want to say is, regardless of all the statistics that we see about which life science cluster is larger, has more employees, has more companies, the thing about San Diego is that it's better positioned and better prepared to take advantage of the opportunities in biotech than any other in the world in the future. And part of the reason for that is what you're going to hear here tonight with regard to genetic sequencing and how we can put it to use in the future to develop products. So as Ivor said, you're going to have the distinct privilege tonight of hearing from some real pioneers who have changed their changed the field of DNA through their work and in building companies that have and will revolutionize the ability of researchers from around the world to produce products faster, more efficiently, more accurately, and most importantly to us to deliver those to physicians and patients. So with that, let me, with great pleasure, turn it over to our co-founder of Biocom, the moderator tonight, our first chair, who's also led some of San Diego's premier biotech companies, including as president and CEO of the one that Ivor started, Hybertech, many years ago, and has since enjoyed great success with a number of companies right up to today. Please join me in welcoming David Hale, who will introduce our panelists tonight.
Thank you, Joe. Can everybody hear? The microphone's on and everything? Okay. So first of all, I'd like to add my welcome to you as well. On behalf of the Life Sciences Foundation, of which I am a board member as well, and also for Biocom, which I am a board member as well, we welcome all of you this evening to what I think is going to be a very exciting panel. Now, most of you that know me know that typically I would have a tie on for an event like this. Well, in honor of our panelists tonight, who I knew would not have ties, I took mine off before coming into the meeting. So, anyway, thanks for that.
You made Joe uncomfortable.
Yeah. Well, Joe wore his tie because he knew I was going to have one. So, anyway, moving on. So one of the things that normally happens at panels like these is you spend 10 or 15 minutes introducing the panelists, and if I were to really introduce them, it would take me 10 or 15 minutes to introduce each one of you based on the significant accomplishments and experiences they've had in this field. So I'm not going to do that. If you want to know about them, go online and you can read for days about what they've done and what they've accomplished. I do want to just say, for those of you that don't know who they are, Jay Flatley to my left here is Chief Executive Officer of Illumina. Craig can't figure out which job he likes, so he's got several. He's Chairman and CEO of the Jay Craig Venter Institute, Chairman and CEO of Synthetic Genomics, and Chairman and CEO of the recently formed HLI.
Human longevity.
Yeah, Human Longevity, HLI. And then right now, Greg is out of a job. As you know, Greg was the CEO of Life Technologies for many years, and that company recently closed its transaction with Thermo Electron Fisher. And so Greg's probably on the street out there begging with, you know, there's people on the street.
I offered him one of my titles.
That's true.
So probably what I would like to start with, as Ivor indicated, LSF is about history and about recording what's happening and what has happened in the biotech industry. And so what I would like to start out with this evening is asking each one of them to take a few minutes and talk about how they got involved in this whole area of genomic research, genomic sequencing, and how, you know, they got involved in it initially. And I'm going to start, Jay, with you and ask you just to talk about how you got involved originally in developing this technology.
Well, I guess my first exposure to life sciences came in 1987 when two friends of mine asked me to co-found a company with them that wound up being called Molecular Dynamics. And before that, I was in the high-tech business, knew nothing about life sciences whatsoever. And I went in to meet these guys, and they had this electrophoretic gel with a power supply and a bunch of wires on the table. And they said, we're going to figure out how to image this. And it was unbelievably unimpressive. I mean, it was like, should I be impressed by this now? And I wasn't. And it took them a couple months to convince me to go do this. And so we put that company together in '87. We spent about, we took it public in '93. We spent the first period in that company's history doing imaging of electrophoretic gels using radioactivity and fluorescence. And then the breakthrough really was when we met up with Affymetrix and Steve Fodor, and we built the first 50 imaging scanners for Affymetrix. And so they were, at that point in time, the dominant array company in the marketplace. And so that was a period about '93 to '95. And then in '95, we acquired a technology that allowed us to build the first capillary DNA sequencer, which a product we launched in about 1997 called Megabase. We tried to sell it to Craig, but he wouldn't go for it. He bought the other one. And the competing Life Technologies, or then ABI sequencer, sequenced about 2/3 of the first human genome. And with Megabase, we sequenced about 1/3, maybe generously 1/3 of the first human genome. And then we sold that company in 1998 to Amersham in the UK, who subsequently got sold to GE. And a year later, after I finished the merger activities, I came to San Diego to become the first CEO of Illumina. And we immediately launched into the array part of the product line using fiber optics rather than the older technology that we had previously used, and then got into sequencing through the acquisition of a company called Solexa in 2007.
Okay, thank you. Craig?
Well, it was definitely in the same decade. I think the 80s is sort of what spawned the field of genomics. I was actually studying neurotransmitter receptors. So in '84, I joined the National Institutes of Health and sequenced the first adrenaline receptor from the human brain. And using the standard gel and radioactive technologies, it took over a year to do this one gene. And it was painful. It was, you had to guess what the sequences actually were from these gels. And it's also right around when Lee Hood's lab announced that they had this new method of attaching 4 different colored dyes to the basis of DNA and could activate them with a light beam and read that into a computer as it ran down a gel. And so we were my lab at NIH was actually the first one to get this working. In fact, in '87, I think we published the first paper of sequencing genes with this technology. But evolutionary biology was moving very slowly, and you sort of did one gene from different species. And so it was around '85. In fact, the Genome Project started with a suggestion here in La Jolla. And I just got immediately excited about it. The notion of sequencing every gene versus the way they were being done one at a time, usually a decade per gene per laboratory, and it is a rough guesstimation. So the rate science was proceeding without genomics, we'd have maybe a quarter of the human genes discovered by now. Maybe not. It was going to be a long, long road because they were done as I was approaching adrenaline with a function that you could trace down and then try and get the gene associated with it. And I just got excited about it, and having the only lab that had an automated sequencer working, I thought, gee, we have the technology that could actually do this. Jim Watson got very excited about it, and it seems pretty ludicrous today when we look back at these early machines. It's like trying to do a 100-mile bicycle race on a unicycle with a flat tire. I mean, they barely sequenced 16 reads per day. But, the notion was, as it happened with Henry Ford and, the whole manufacturing scenario, one's not enough, you buy a whole bunch. And it led to, in '87, being annoyed with the slow pace that the government was moving. It wouldn't have finished even this century at the rate it was going. So in Applied Biosystems introduced its version of the capillary sequencer, which actually worked. That's why I'm in the middle here.
See what we deliver you next week.
But I've changed now. They've improved. So we bought 350 of these and set up a giant factory. That led to the first draft of the human genome. And but it's just getting started. In part, you know, the only thing that's really changed in the last 15 years since we sequenced the first draft is the technology, and it is really stunning. So we had this building with 350 of these instruments. It took 9 months to sequence the first draft of the human genome. And the technology has progressed. Illumina in particular has, with their announcement of the X10 sequencer, has a machine that I wish the hell I had 15 years ago, that we're scaling up now with 2 of these units to do 40,000 genomes a year versus 1 in that same time frame. And you know, life has developed some exciting technologies as well, and you know, that's the value of, you know, the San Diego community. It's now the home of this entire industry. And right after we announced HLI, I got a call from a Boston Globe reporter saying, so you're telling me the center of genomics has now moved to La Jolla? And I said, you're just recognizing that in Boston?
Yeah, they're slow to come around.
Yeah.
They're in denial.
They're in denial for a while.
So Greg, what about you?
You know, I maybe tell the untold story of our journey. In 2006, we were just coming off of consolidating a number of reagent companies. We were Invitrogen at that time. And I got a unwanted phone call from a gentleman named Tony White, who Craig and I know and have history with. And he was the chairman and CEO of Applied Biosystems. And he said, we want to do a takeover of Invitrogen. This is kind of the untold story. I said, well, we're not for sale. We're a little busy right now doing all this integration. And he was very persistent. And what ensued was 3 to 4 months of just a total headache trying to not have to deal with that situation. And Jay can appreciate that from his own situation with Roche a few years ago. And that went away. And then the next event was, Jay, you may remember this. You and I were flying up together on a plane, and we started talking about sequencing.
Yep.
And I think you had just acquired Solexa, and quite frankly, sequencing for us was a bridge too far. We were just a consumables company at the time. And I remember just thinking to myself, wow, that could be a really big opportunity. A year later, a hedge fund manager called me out of the blue, who's now going to jail, SAC Capital. It's all in the headlines. And he said, Greg, why don't you take over Applied Biosystems? And I said, that is an excellent idea, actually. And so we did. And that's how we kind of backed into being in the sequencing business, was taking over Applied Biosystems and then renaming the company Life Technologies. And Craig and I have had great laughs since then of Tony White stories and what we had to deal with and not deal with.
He fired him. He's my hero.
So if you look back over the last 25 years, I think history will certainly report that, the sequencing of the human genome was one of the most important events in that period of time. Maybe, Craig, you could talk for a minute about that race and about, what the environment was like at the time, and it was a real race, and so.
We tried to deny that it was race, but, it kind of was. But we made it a race, and it, the backstory behind it is interesting. Somebody read about it in my book, but I kept getting these calls from Applied Biosystems from a guy you all know, Steve Lombardi, who said, we want to give you $300 million to use our new instrument to sequence the human genome. I thought they were crank calls. I just hung up on the guy. And finally, he flew out to meet me and said, no, we're serious, and you have to come out and look at it. And so I flew out with a couple of my colleagues to Applied Biosystems, and they showed me their breadboard device. And it was actually, their sequencer was separated into 3 different buildings. But it was clear, Jay clearly had the same vision. Switching to capillaries totally eliminated this problem with these slab gels, which were very inconsistent. And from the raw data, it looked like they could actually put something together and it would work. And so I kind of hoped they were serious and they were just going to give me the $300 million from my institute. But these capitalists, they have different visions of things. And so they insisted on starting a new company, which we named Celera. And it was to use this new capillary sequencer to sequence the genome. And you know, it's not the kind of thing you could keep secret, and Celera became a tracking stock, so we had to announce it and deal immediately as, you know, so I became this instant head of a New York Stock Exchange company, which is not a position I would ever take again if I could avoid it. And so we had to announce everything, and the announcement kind of took the world by surprise, in part because everybody had this notion that sequencing the human genome was an impossible task. And if you're doing with radioactive material and these slab gels, it probably was. But the automation that started with Lee Hood and then developed into all these new technologies, you know, changed that conceptually from the beginning. Putting it in practice took a long time. The government labs were actually not serious about getting it done. And there was always this battle between the genetic mappers and the people like me that just wanted to get on with sequencing chromosomes to move it ahead. And I think if Celera was not announced and the race was started, it probably never would have gotten done. And a lot of people, you know, if you really look at it seriously, it probably never would have gotten the rest of funding from Congress to be finished. So you know, sometimes competition is really good, and it did spur things on. When there's this kind of competition, the best thing to be is the business these guys are in. You want to be the arms dealer. And Applied Biosystems sold weapons to both sides as fast as they could. So they made several $1 billion off the race to sequence the human genome. And I used to be annoyed my own company wouldn't even give me a preferential service treatment because they were out trying to get new customers. And so their secret plan was just to sell instruments. I actually wanted to sequence the genome. And we did, and 9 months later, it was finished, and I got fired. You know, so the competition was more because of these preset ideas in academia. And it was not, you know, biology's never been scaled up before. And that's what both these guys have been responsible for is, you know, scaling up and automating biology and really making it high scale, high throughput. And, you know, physicists caught on to this a long time ago. Biology is treated as something you do in your little tiny lab by yourself with 1 or 2 people, and you don't do it on an industrial scale. You don't do it in an integrated fashion. And I think that's what genomics has changed for science. It's made it an integrated science, and with more data now than almost any other field is generating. And the biggest challenge we're dealing with, and we've bought 2 of his $10 million instruments, as you know, to scale up sequencing to a level that has not been even imagined before, is the data processing side is now the challenge versus generating the data. And what Jay and Greg and their teams have done, I totally applaud. I'm the consumer. You know, I don't build instruments. I keep these guys in business by buying them and trying to take it to the next stage. But that's why we have this technology advantage in the US with all this stuff that can't be done anywhere else.
Okay. So, Jay, just focusing on where the technology is today, what are the applications that you are seeing it being used for primarily? Today, not in the future. We're going to get there in a few minutes, but today.
Well, the research market continues to be a very robust market, even in the face of the challenges in funding in the U.S. and to some limited extent other parts of the world, and represents over half of our business even now. I think probably 3 years ago, maybe 4 years ago, the shift began to take sequencing technology into the clinical setting, and this has now become a very major part of our business. But it took some changes in the underlying technology, some improvements in software certainly, in sample prep, increased automation, and the fundamental research that had happened before to make the clinical utility of the genome useful in a clinical environment. And specifically, I'd say in the last couple of years, there's been maybe 2 major segments that have developed extraordinarily fast, perhaps faster than any of us thought. One of those is a company called Myriad, which uses DNA sequencing to sequence the BRCA genes, and we all know about Angelina Jolie's story, which sort of brought that forward with great publicity recently, but that's been going on for years. And they're moving to next-gen technology. They were using prior generation technology up until recently. But probably the most interesting story is what's happened in the non-invasive prenatal testing market. And this began really only 2.5 to 3 years ago with the discovery that by doing deep sequencing and counting in maternal blood the fetal fraction, so the presence of DNA from the infant in mom's blood, that you could actually replace amniocentesis and detect Down syndrome and other syndromes through that method. And this, looking back on it for obvious reasons because of the invasiveness of doing amniocentesis, in the course of really 2 years of full marketing of this technology, over half the women in the US now in the high-risk category opt for sequencing over amniocentesis. So it's probably the fastest adoption of any diagnostic test that I know of. Certainly, usually these things take 5 years or 8 years to really get to be mainstream. And just recently, there's been the first publications that this is equally as useful in the low-risk population of women. So it works just as well in the low-risk population, which historically hasn't been a target for amnio because the risk of the test outweighed the advantage of doing the test on someone who's in a low-risk category. So from a clinical perspective, those are probably today the two biggest subsegments, and then lots of broad use in other areas in cancer and other techniques.
Okay. Greg, I'm going to ask you to comment on that. But first of all, we've got lots of people standing in the back, and I know you didn't want to interrupt. So we're going to take a second for a break and ask you all to take a seat. There's some seats all the way down front. Some seats over here. Okay. So please come on. We'll wait for you. All right. So with that, Greg, maybe I'll ask for you to comment on that as well.
Well, I would just add to what Jay is saying. I think the other area that's now just emerging in terms of a viable business is in cancer testing beyond just single genes or small sets of genes. And it's really being driven not only from a technology perspective, which we're talking about tonight, but two other big trends that have happened. One, just the plethora of cancer drugs and being so expensive that they are. And then second, the desire by all sorts of entities, employers certainly, to capitate their healthcare costs. And cancer drugs, cancer patients are one of the big cost drivers in healthcare. And so I think you're going to see ever more use of sequencing tools to really drive therapeutic selection in cancer, and that'll be one of the next big markets over the next couple of years here.
Okay, and then Craig, any additional thoughts?
Well, the present is a rapidly moving window,
the present.
Right? So
today.
The present today, we're waiting for instruments to be delivered.
I promised him tomorrow.
He said manana.
Won't fit in the back of my car.
So tomorrow we will be getting our first X10 delivered to start this scale up to first 40,000 human genomes a year, and then hopefully up to 100,000. And so today is trying to build this infrastructure. And I've been saying for almost a decade, the challenge would not be sequencing. It was clear that it was coming down. It wasn't clear which technology would get it down the fastest in terms of cost, but it was the other things around it that were going to make the difference in terms of whether it's cancer or any other diagnostic, and that's characterizing the human phenotype. So we have whole teams trying to do that, of defining what it means to be human and where it can be completely digitized. And so we're layering on the genome with the microbiome, with 2,400 chemicals from the metabolome, with the proteome, with this complete human phenotype. And the compute resources around it are, everything's just on the verge. I think Jay and Illumina pushed things just past the threshold that I was looking for, this sort of magical $1,000 genome mark. You know, because the cost of doing our first genome, you could do almost 100,000 genomes, right? It's a very different world. And if you take the federal program combined with the British government and the Wellcome Trust, spent about $5 billion for the first public draft. That covers a lot of genomes at $1,000 a genome. So the data side is coming quickly, and it's now ready to become, in my view, and that's why we're pushing the limits here in San Diego with the Moores Cancer Center with our announcement. We're going to do every patient that comes to the Moores Cancer Center, and we're going to do full genome sequencing on their tumors as a start. And there's only about 5,000 patients that come through the Moores Cancer Center, so it's only going to be a small portion of what we do. But I think the other argument I've been making for a long time, genomics is the law of large numbers. I've had my genome now on the internet for almost 15 years. When it was first sequenced, there was nothing to compare it to, and you could almost discern nothing from it. If we have a database of 500,000 to 1 million, as sort of the starting point for comparisons, we will be able to get people very accurate with minor error bars on statistical significance of what the different traits are going to be. And we were talking about earlier just this report that came out 2 days ago for forensic uses of predicting somebody's face from their genetic code. It was really stunning to me, even at the level of data they had. Those of you who saw the photo, it almost looked exactly like this woman's photo. So we're going to be, that's going to be part of our phenotypic characterization. But just think whether it's from maternal fetal cells circulating in the maternal bloodstream or other tests, you could actually predict from that somebody's complete genetic repertoire, their susceptibility for different diseases, and different key traits, even potentially what they're going to look like in the future. So to me, this is what I argued when we were sequencing first, that this race that you referred to, I called it a race to the starting line. And I think with what we just announced with HLI, we're now at the starting line. And this is going to revolutionize medicine in ways that everybody's dreamed about, but can't be done without the large numbers.
Okay. So
a little bit of future in there, but not a lot.
I gotta,
He's dabbling.
it's really hard to keep him in the present, for sure.
I'm not a head of a public company. I can speculate all I want.
Me too.
Yeah, hey, that's true.
So
He thinks his stuff will be good.
Yeah, I can't say great.
So each of you had your genome sequenced. And Craig, like you said, you put yours out there, 15 years ago, and you talk about it in your book and so on. But maybe if you're comfortable, what did the three of you find that was the most interesting when you had your genome sequenced in? Would you do it again?
We sequenced in 2010 Ozzy Osbourne. And very interesting genome. Had an overexpression of a gene that produces the enzyme in your liver to accelerate the processing of alcohol. And that's why he's probably still around, right? And so when I got sequenced, I had that gene, fortunately, so that's good. That makes for a good weekend.
I've seen that in action.
I think more in a sober way, though, was I also was getting sequenced just after we had done my mother. And we're early on in this. We were doing my mother because she had had a freak accident, hit her head, and then a few months later developed symptoms of Parkinson's. And it was very unusual, so we sequenced her, and it turned out she did have the genes that are known to be associated with Parkinson's. And so it really spurred for me a desire to better understand myself, and that's why we then went and sequenced myself. And turned out I had those same genetic markers as well. It's very interesting, though, because a lot of newspapers were writing articles about that lineage of my mother to myself. And we went on and then did my daughter. And but the one person I couldn't get was my father to do it at the time. And so there was a lot of discussion about, what does this mean in terms of preordaining your future and destiny and the like. And I think most of the media got it wrong at the time. For me, it wasn't anything in terms of, a certainty. It wasn't in any way negative. For me, it was liberating because I could participate a lot more in terms of clinical trials, one of which I'm involved in right now with Parkinson's disease. I could be more of an emissary about how this allows you to be more knowledgeable and aware of what you can do to make a difference with your years. And I just think it's just so revealing, and in many ways, it is what life is about, that the more you know, the more you can do. So, those were the two things that we found out when we did myself.
Jay, anything you're willing to share?
Yeah, sure. So I was done about 4 years ago when we were launching our CLIA program for beginning to do this at a consumer level, and we needed I call it a dummy genome to go through the lab the first time to get the CLIA approval. So I was selected, and so I got sequenced, put my genome in the public domain. And one thing people don't always realize is that if you sequence somebody who's generally healthy in sort of the 50s or 60s, you're unlikely to find things that are catastrophic in most genomes. We've now done hundreds of people at the consumer level, and we find sort of 2% that have something that's really that they ought to go see a doctor soon about. But in my genome, we found a couple things. I was a carrier for a couple diseases, Wilson's disease being one of those. I had a childhood syndrome that nobody knew what it ever was, but I used to get a very severe rash in school when I got cold, and turned out that was something called familial autoinflammatory cold syndrome, which I knew nothing about, but we discovered it through my genome, and the world's expert happens to be here at UCSD. But one of the real powerful things about getting sequenced is you don't have to do it again. And so what happened, every year we run our genomes back through the brand new pipelines and the new clinical pipelines that take advantage of the recent literature and discoveries. And so just this year when we did that on my genome, we discovered that I have malignant hyperthermia. I have the most powerful variant for malignant hyperthermia, which I didn't know what that was either. Turns out that this is the variant that causes people to suddenly die under anesthesia for no reason. And you often hear this, where they're on the operating table, all of a sudden they just die, and it's not because anything went wrong with the surgery, and this is the condition that causes that. Critically important because it's an actionable result of the genome, because if your anesthesiologist is aware of this, they can prescribe alternate anesthetics, and if they get this calcium rush, which is behind this, they can fix it if they're ready to go. So that's a great example, we think, of something that's highly actionable in the genome.
And Craig, for those people that haven't read your book.
Yeah. Well, one that's of more recent interest that's helping to drive some of the things we're doing. So it's been out there. Jim Watson deleted it from his genome, but I'm heterozygote for the ApoE4 allele that gives an increased risk for Alzheimer's disease. There's no history of Alzheimer's disease in my family, but it's also hard to tell on my father's side. My father died at age 59 from sudden cardiac death, so he never lived long enough to find out if he would get Alzheimer's. And so, as Greg was talking about with this increased risk, you think about it quite a bit. And I decided to take part of a N of 1 study here at UCSD with this radioactive dye for detecting amyloid in the brain. And the neurologists were so concerned they made me do it under a pseudonym. I told them my name was Greg Lucier.
No wonder I can't get insurance now.
And because they were worried what the consequence would be, because actually the neurologists were assuming because of this increased genetic risk, they were sure it was going to come back positive, in fact, very positive. But see, that's the trouble with the existing genetic test. When you hear you have a 30% increased risk for something, and it even started back at Celera. We said everybody looked under the lamppost for their lost keys because that's where you could see. And that's what's happened with genetics. People looked at traits they could track down. And so they look at associations with disease. They don't look at why you don't get the disease. So basically, I have a clean bill of health for the next 20 years, at least with amyloid-based Alzheimer's disease. which was a real relief. And it's actually, it turns out, that just a little bit of publicity about this, a lot of people that are ApoE4 heterozygotes now want to get this test, because what the neurologists say is that people that think they have these increased risks start behaving or worrying about their outcomes as though they do have that disease. This happened early on with the first test that could actually be done with Huntington's disease, right. And it turned out a lot of members of families with Huntington's disease present didn't want to know, because in fact their entire lifestyle was built around maybe having the disease, and they didn't want to be isolated from their siblings who might get it or might not. So there's some cases where people actively choose not to know. But it turns out people that have this increased risk for Alzheimer's disease start interpreting every little absent-minded thing that happens as we get older with, these are obviously symptoms of Alzheimer's disease coming on. And I worried about that, and I bought a faster motorcycle as a result. So it was actually very liberating to find out that I didn't, but it also raised more scientific questions. And so that's part of the goal with doing these really large numbers. Right. We want to find the protective alleles in the population. There's a reason it's only 30% and not 100%. And everything in the genome, with a couple of exceptions, is statistical probabilities. There's very few yes-no answers. As we narrow the question down, like with the ALK gene and lung cancer, you can start to get yes-no answers. But the broader, more complicated traits are statistical probabilities. But we can actually change that. If we know the protective alleles, we can say, well, you have these 6 protective alleles and you have these 2 risk alleles. Here's the exact statistical data that comes out from looking at this in large populations. We can tell you which category you're in unequivocally. But you can't do that with a single risk allele. So we need the large numbers, and we need these large patient outcomes. And the only way genomics will really have a big impact, other than these one-off gene tests in cancer and a few other things, is if we get these large population studies. And San Diego is now the bleeding edge of that entire movement.
Okay. So how many people in the audience have had their genome sequenced? Boy, there's a big opportunity out there. So another question.
Humanlongevity.com.
Yeah. So would you, the 3 of you, and maybe whoever wants to start, would you recommend that a mother, either prenatally or after the baby's born, have the baby's genome sequenced?
The full genome sequence?
Yeah,
not
full genome.
Well, I guess we always say at the end of the day, those choices will wind up being personal choices, so we don't recommend one way or the other. I think if you're actually talking about sequencing the entire genome of a fetus prenatally, there are clear ethical questions that arise as a result of that. And so as a company, we've taken the position that we don't offer such a test, even though that is technically feasible today. We don't sell that. If somebody bought our instruments, there's nothing we could do to prevent them from using our instruments in that way, but we don't do that work.
What about after the baby's born?
Well, it's a different situation after the baby's born to some extent. I mean, the question then is, is it correct to sequence an infant, let's say today, with the clinical utility we know of in the human genome today, without the consent of that individual? Some people would argue yes, if you have enough reason to do it. Either there's some specific medical condition, which we do quite broadly, and not just we, all of our customers, all of Life customers are involved in sequencing rare diseases in children under the care of a physician. The question becomes, if the baby is a healthy, apparently healthy baby, do you allow that baby to be sequenced? Today, we do not do that unless it's recommended by a physician for a specific reason. I think in the future that will change, and it's very much sort of like the analogy of the internet. In the early days, there was more worry about typing your credit card number in the internet because of the bad things that could happen, and the advantages weren't very important at that stage. Where today, you know, the advantage of doing that overwhelmed the risks, and I think that will happen as we learn more about the clinical utility of the genome. For example, if we can detect Alzheimer's on the second day instead of when a child is 7 years old, if you know, after the kind of work Craig and others are going to do using these technologies, sort of what's behind these diseases, and you can detect them early and actually begin treatment, then the clinical utility would be justifiable.
Craig?
I think I'm a little bit more on the bleeding edge of this. I think the time is now to absolutely do this, and not only on the fetus, but preconception. You can actually predict outcomes before a couple gets together and has sex to produce a child. You can predict very important things in advance before that ever happens. And the trouble with these rare alleles, how do people find out? They have a child with an extreme deforming disease. One of the best examples is with ataxia-telangiectasia. Brad Margus, who I think everybody here knows, was a Harvard business graduate, but he had a shrimp farming business. He had 2 kids with this very debilitating disease. They sort of die in their late teens. And he started a foundation to fund the research to find the gene associated with this. Right. And actually benefited from that himself using pre-implantation diagnostics. Had 2 healthy kids because they could select for cells without the defective gene. And it's a rare allele, and it's a dominant allele, and was basically even unknown at the time. All these can be prevented in advance, but if couples know they're even at risk for it ahead of time, you're set up to then do these tests. Maybe that's what drives it forward to doing these tests. Information is power. Knowledge is power. If you have lung cancer, you need to know whether you have this mutation in the ALK gene. If you don't have that knowledge, you're subject to random medicine, right, and the likelihood is you're going to die from lung cancer. So it starts at the earliest stages, and as I said, even preconception, that knowledge can be very powerful.
Okay, Greg, any comment?
Well, I would just add, I think they've done a nice job bookending the dialogue. What you are seeing in the marketplace today, though, is just an incredible energy to provide ideas and ultimately choices for the patient, for the parents. Boston Children's is trying to do every child coming in through the front door their exome. And they're partnered up now with Cincinnati Children's to do the same. There are companies now that are starting to offer the service that Craig is talking about. And so I think over the next couple of years, the choices that the patient and the parent are going to have are just profound. And it really raises the larger question of how do you make the right decision for yourself, for your child. And I think those are some of the big questions here for the next few years.
The Chinese government just banned genetic testing in China. They think it's a moratorium, not a complete ban. And the reason for it is it's been the #1 use of genetic screening in China is sex selection.
Yeah. Right?
And they're getting way too many men in China, so they, but it's really a problem because, with all the instruments Jay sold into China, genome sequencing's become a big deal there, and with the one-child limitation, genetic selection of all kinds has become very powerful and prominent.
No question.
And so now the government's taken the complete opposite tact and banned it everywhere. But, that is where the ethics become problematic. None of us want to see this technology used for sex selection or other trivial selections. When you're dealing with a disease like ataxia, it's totally different, and the community's pretty unequivocal about that. And so one of the polls that was done early on in the genome race done by USA Today asked parents if they could alter the genetic code of their child to have more positive traits, would they do it? And way over 60% said absolutely. So I think, the technology and the basic understanding of what it can do and what it will be used for, we have to worry about what Jay was talking about, the moral chasm out there that could develop of people not understanding the importance of what they're using it for.
And I might clarify to your point, Craig, that my personal position on this is different than our corporate position. I mean, I sort of come from the position personally that everybody should get sequenced and put into a database, and we should build these, and that's how we're going to create great science and cure large fraction of human disease. From a corporate perspective, we have ethics committees. We have to be very delicate in terms of what we actually go out to the market with and say, we're selling a test that does this.
Well, and we have to get FDA approval for all this stuff that we're doing. And can we disclose that you invested in HLI?
I think you just did.
Okay.
I think that's public, yeah. That is public. And we, by the way, we have a business in pre-implantation genetics where we actually do a genetic test on all of the embryos and select the one that doesn't have the aneuploidies for implantation. So that's a business that exists today.
So let me propose that something and see what your thoughts are, that sequencers will never move to the doctor's office, but the information will. Do you agree or disagree?
Well, you totally agree on the latter, and that's already started to happen. The former, I think, will depend on the cost, the simplicity, and ultimately the need, if that makes the most sense. Yeah. Quite frankly, if you look at the continuum of diagnostics, while point of care has been an important part of the overall diagnostics industry, it's not the biggest by far, right? It's never achieved its potential. And my gut would say sequencers will be no different. I suspect they'll remain back in the hospital lab, back at the reference lab, and sporadically in certain situations you'll see them out at the fringe in a doctor's office. But most certainly, the genetic report will become a really critical component in the overall diagnosis and understanding of that patient in front of a doctor.
I think the one exception, I agree with you, Craig, by the way, is that I think you'll start seeing them in the ER in the next 3 to 5 years. They'll be small enough, easy enough to use. The run times will be fast enough that a patient will come in and they'll get sequenced right in the ER. And there, it actually can change actions quite quickly.
Craig, any? Okay.
Smack dab in the middle here.
But I think that all of you would agree that information will become a part of clinical decision processing. What is it going to take in order for that to happen? At the doctor's office level, where most of us...
Yeah. I was chatting with somebody the other day. I was very impressed just last week. We had a meeting at Sanford Burnham with a number of children's hospitals in Sanford Burnham, and we were talking about a larger program we would do together. And one of the presenters was Sanford Health out of Sioux Falls, and Denny Sanford had been very gracious to them and gave them $100 million for a clinical genetics program. And the person who stood up was an excellent, excellent leader and talked about how they were going to transform the Sanford healthcare system to integrate genetic information into their actual care of patients. And one of the things that you haven't seen really happen yet in the healthcare system is the rise of the genetic counselor. A lot of people have talked about it, you hear it here and there, but theoretically, if the future is what we all think, the genetic counseling business should be huge. It should be as big as anesthesiology.
Right.
And so I think it will. And I would just simply say Sanford Health was the first healthcare system that I thought had the vision to start building up the genetic counseling effort, creating a program to train them, and integrate them into their overall clinical care at all their hospitals across the Midwest. So I thought that was really foresightful.
I think the big breakthrough that has to happen is that, and it is happening right now, is the connection between the genotypic information and the phenotypic information, exactly what Craig is starting to embark on, and others around the world at smaller scale are embarking on as well. And these data sets, as they get generated at the kinds of scales we're talking about, hundreds of thousands of genomes, the clinical application for a physician will accelerate if we can figure out how to share the data sets. And I think That's something that lots of people are working on. Are there more common ontologies and database structures that allow these types of data to be combined? Because that's where you get exponential power from collection of the information. And we do envision the day where the physician does a query that says, here's the sequence of the patient I'm treating. What are the 50 most similar ones, and how were they treated? And that's going to happen.
So the thing that gives me encouragement that this could happen relatively quickly is, as Jay referred to, the last 3 years of what's happened in cancer. The information finally became actionable and meaningful, and it got incorporated almost immediately. And I think that's what's going to happen with this next stage, because with the larger data sets, we're going to have, I predict, a couple exponent increases in the number of actionable bits of information we will get out of the genome. And as soon as it is actionable, and as soon as you send your genome or your child's genome to our database, you get back a report right away that says, here's all the different factors that you should take into consideration. That instantly makes medicine totally an information-driven practice versus the kind of subjective practice it is today. And as soon as that data is meaningful and actionable, like the ALK gene, I think it will be incorporated. But how do physicians learn today? And this is not to insult physicians. Most learn about the leading edge of medicine from their patients, not something they learned in medical school. So we have to start incorporating genomics and this new information wave that could overwhelm people, but that's what these new computers are going to do. We don't have to rely. I'm bothered both on the potential need for genetic counselors, and I would hope in the long run we can do without them, that the computerized information will be powerful enough to give physicians and families sufficient information that it becomes readily apparent. The reason genetic counselors are needed today is the information is so limited that you sort of have to understand the odds of different traits showing up in a family, if it's a dominant trait versus a recessive allele, and you have to understand genetics. Genomics is not genetics, actually. That's a different part about it. I'm not a geneticist. I hated genetics. It was really boring. Genomics is taking it to a whole new level of information science that, Nick Short, who's on our team, has been using this term N of 1. But everybody is an N of 1 when it comes to genomics. And, we sequence several sperm cells. No two sperm cells are alike. No two egg genomes are alike. That's why it comes out different every time. And so what's standard for medical practice is looking up these tables of what a normal value is meaningless when it comes down to you. Larry Page, the head of Google, said if we eliminated all cancer, it only changes the average lifespan by 3.5 years. But averages are meaningless if it's your lifespan. Right? Mm-hmm. And that's what N of 1 is about. And that's what this information is going to change. Greg now knows he has this risk of Parkinson's disease. He can start to watch out for it and study changes. I did this with Alzheimer's disease. What's happening at the forefront of this area? Whatever people are susceptible to, they're going to be much more attentive to, and even help drive new treatments associated with things. So the N of 1 is probably the most important concept.
So you mentioned earlier, one of you mentioned microbiome, the metabolome. How is that, epigenetics, pharmacogenomics, and so on, going to be all integrated? And how is it going to affect this information that we're going to have?
Well, if I can take this first. All these different areas are sub-disciplines of science. And they get, they have different meetings. So the people who study the microbiome go to microbiome meetings. The people who measure the chemicals in the bloodstream go to these metabolome meetings. They don't care what your genome is. They don't care what your microbiome is, It has started, I was driven by my early years here at UCSD. Early 70s was the era of cyclic AMP. Some of you guys remember this, where every paper in the scientific literature was some key physiological event in the human body driven by cyclic AMP going up or down. And I said, there has to be more to life than this, right? And so we're attempting now to integrate all these levels of information. As I said, the technology is just at the threshold, and the computers are just at the threshold for trying to do this. And that's what we have to do. We have to integrate knowledge. And there's no better place to do that in San Diego. This is where the original multidisciplinary programs came out. People actually talk to each other across the mesa. And so integrating these disciplines, integrating all this different data is key. We should be able to predict from your genome which microbes you're going to have, which diseases you're susceptible to in terms of infectious disease. And there's an argument amongst the people who measure proteins versus chemicals. The people who measure chemicals say they can predict what the proteome is going to be because these are the metabolites. And the people who measure proteins say, well, we know what the chemicals are going to be because here's what's there. But we'll find out what's actually important. We work as integrative systems. We don't work on just our microbiome. Sometimes these things take more or less significance, but that's what we're trying to do. We're trying to use it to put it all together.
David, just to add, though, I spent my career at General Electric before I came out to San Diego, and in the diagnostic imaging field, if you go back into the early 70s, late 60s, there was basically only an X-ray machine. And you had radiologists out there, and yet they weren't that important, and there weren't that many of them. The rise of the radiologist, of that subspecialty, was primarily driven by the technology of the CAT scan, of the MRI, of the PET scan, And all of that sophisticated imaging system required an integrator and a human to look at the images and then translate it, again, just to a doctor, right? Because the doctor ultimately then was who the radiologist was talking to. It may be a computer, and I want to be respectful of that, but my sense is for a while in our healthcare system, somebody, a human, whether it's the pathologist or a genetic counselor, somebody's going to have to be the integrator of all this information. And I think we're going to see the healthcare subspecialty change in some way to handle it.
I think from a technology perspective, whether you're trying to measure the microbiome or the metabolome or the epigenetics, sequencing can get at most of this. We have a ways to go in areas like simplifying sample prep and increasing read lengths for things like the microbiome. So there's continuous improvement that needs to happen across the board here, and obviously price, Craig continually reminds me. But I think that the basic fundamentals of the tool set...
Anything you multiply by 100,000 becomes a really big number.
Really big numbers. But I think the fundamental tool sets today are so enabling compared to where we were just 5 years ago. And we were woefully underpowered 5 years ago in terms of tackling these problems. And I think today we have it at the disposal of people like Craig, the tools to go get this information and integrate it.
I think Jay's more right than people probably want to accept, that the genetic code is actually going to be the predictive element. And people always say, what about epigenetics? And I want to issue T-shirts that say, epigenetics are still genetics. Everything that people measure as epigenetics originated with the genetic code. That's what we proved with the first synthetic cell. We just put in synthetic software and everything derived from that. And so ultimately, if the databases are big enough, we should be able to predict almost everything from the genetic code.
So something that I know is interesting to a lot of people in the room is how is NextGen sequencing going to affect drug development in the future?
Well, I think it's beginning to have a pretty big impact now. It's been used historically, in limited ways for drug development. Back in the 90s, the large pharma companies were all doing large-scale sequencing programs for target identification. I think you've seen over the past couple of years, after a lull, some resurgence in the use of sequencing by biotech and pharma companies. Regeneron announced recently they're going to sequence 100,000 exomes, which is kind of a shocking thing to hear them say. Decode in Iceland has imputed the sequence of most of the Icelandic population, and They were bought by Amgen for $450 million because of the database that they were able to create. And so I think in target ID and in enriching clinical trial populations, in beginning to sequence the clinical trials themselves, I think we're right on the cusp of that beginning in a serious way. It's actually been a real surprise to us that pharma didn't do this earlier. From a scientific perspective, we could have been running microarrays on every patient in clinical trials 10 years ago. And when you really probe under the covers to figure out why that was, the reason was that they were afraid of creating data that they couldn't explain to the FDA. And I think that's been an underlying problem in the use in clinical trials. And I think that's the new FDA, if you will, is beginning to realize that NGS really does create that level of uncertainty, and they have to figure out how to live with it. And so I think because of that willingness to live with uncertainty of data you can't explain, pharma's beginning to adopt it again at large scale.
But it was also fear of market segmentation. So
back then it was.
Back then.
Now it's not.
So the Pfizer drug that works on these ALK mutations in lung cancer, the clinical trial failed. If you knew that it worked on that subpopulation, you could have done a very small clinical trial very quickly.
Right.
And so what's changed is the definition of a blockbuster. A blockbuster used to be a drug that was sold very broadly and therefore had broad market acceptance. And now a blockbuster is one where it costs $9,000 to $10,000 a dose for treating a small number of patients in these small genetic subpopulations. So that's what's finally, I think, driven it to be acceptable in the pharmaceutical industry to consider using genomics. And it should totally change how clinical trials are done, the speed and the cost, which means a lot more things can go through clinical trials and hopefully shorten the time for getting new drugs approved.
And success rates will hopefully go up.
Yeah.
So let me ask you the same question about diagnostics.
Well, I think we talked earlier about these different market segments that are unfolding as time goes on here. And maybe just to take your question and go a little farther, will a whole human genome be a standard diagnostic in the healthcare system 5 years from now? It's a great question. My gut would say probably no, but I think if you extend the timeline out a little bit farther, then I would probably say it will be. And
I'm hoping he's wrong on that timeline.
Jay?
Well, I think the use of sequencing has been in the diagnostic world now for probably 15 years at one level or another. I think NGS has really just sort of put an E in the curve in terms of the adoption rates because we've discovered more. We can implement it in much simpler ways at lower cost. So I think the uptake in diagnostics has been quite broad, and probably a third of our business now is driven by some diagnostic use of our technology. And so I think that is really the up-and-coming brand new market. And we're just, frankly, scratching the surface on how this is going to get done. As we move into talking about sort of future applications, I think there's some enormous opportunities in the diagnostic field using NGS.
That's a good segue to my next question, and that is, I'm going to put all three of you on the spot in terms of, thinking ahead 10 years. Okay, where will NextGen sequencing be in 10 years?
In terms of the application or the technology?
The application, not the technology, the application.
Well, it's almost hard to fathom what could be happening 10 years from now, because there's amazing things that are going to happen in the next 2 to 5, and maybe I'll answer that and let Craig go out 10. But,
yeah.
he loves talking about the future. Greg talked about oncology, and I think oncology is probably the next major market that's going to be massive adopters of NGS technology, and across the spectrum of cancer management, from diagnosis to therapy recommendation to measurement of minimum residual disease. Right. I think the most exciting thing that's happened just in the last maybe 6 to 12 months has been this discovery of cell-free DNA and cell-free nucleic acids. And the way this science is progressing is frankly breathtaking. And so, it's now known that some cancers, or maybe lots of cancers, shed their DNA into the bloodstream. And simply by sequencing the blood and counting it the same way that it's done in these NIPT tests, we and everybody else doing NIPT are discovering cancer cases in the moms, where the babies are fine, but the mom has cancer, using exactly the same test. And the statistics are showing that we're detecting a very high percentage of these. And so it really does point to the fact that at least in some cancers, we'll be able to simply do screening tests from the blood and pick these cancers up. Unbelievable market opportunities if that really happens. So, you know, who knows where that'll be in 3 to 5 years, but, you know, I'm ready to get sequenced now. I have my blood sequenced to, you know, to detect cancer early. And this is, you know, you can do this at very reasonable prices. And so I think there's, in fact, there were some people talking in the lobby before this about people working in the cell-free area. Craig's going to be working in this area. We're doing research. Everybody's doing research in this area. It's going to be explosive.
Craig?
In 10 years, at least in San Diego, you won't be able to go to a hospital or clinical center with the first question that's asked is, do you have your genome done? And if not, it will be a requirement before anything else happens. And the question is how fast it will spread from San Diego to the rest of the world.
Okay.
I think in 10 years we will have restored an extinct species using sequencing as the start. And I think sequencing, as well as other scientific tools, the omics, will be a tool by which elite athletes are assessed, judged, and determined.
They are now. It's called the Olympics.
Yeah.
And it's a genetic test.
It is a genetic test, you're right. But it'll be formalized.
But they're not buying a lot of sequencers yet.
Not by sequencers yet, but it will be.
Okay. So we've got a few more minutes for the session. I'm going to take some questions now. Gentleman over there.
I just wanted to ask, I know I've been told San Diego is the emerging headquarters for genome sequencing, where big data meets big biology. Clearly, the big biology part is in place, but I'm wondering if resources exist in San Diego to provide the big data part that you're going to need.
Good question.
Yeah. So one of the most important things that UCSD has at the Supercomputer Center is it has the fattest data pipe in the world. And that's going to be one of the most important things. The biggest challenge that we have is actually moving the data fast enough from one place to another. If you could do that, it really doesn't matter where the computers are. They don't have to be in San Diego necessarily. And one of the first things we've done is being installed right now is we're establishing one of those fat data pipes connecting to the Supercomputer Center where the major switch is out to our facility with all these Illumina sequencers. So we can move data around. You know, there's this, I think Greg may have been the one to coin the term the race to the bottom for the cost of sequencing, but that's what's happening in computing right now. And in cloud computing in particular, you can see Google trying to break in has just dropped their prices again. So the cost of the computes, which is sort of the hidden cost of genomics, you know, we know what the technology costs. We don't really know what the true compute cost is going to be. But as I said, that same technology is just on the verge of being ready. And one of the biggest limitations in computing has not been storage. That's been changing very rapidly. It's actually this pipe for moving data around from one place to another because the size of the data files have increased so much. We had this special fiber between my institute in Rockville and the one here in La Jolla. And now we send data by FedEx because it's much faster because the data is there's so much of it that you can't get it through the pipes. But with these new 100 megabit a second pipes, and it does make it possible. And we're talking to the biggest compute sources on the planet. And it's not just the computers. It's, you know, applying the right machine learning algorithms, the right techniques, the ability to integrate all this data that nobody's ever tried to integrate before. So it's, you know, the thing that allowed us to sequence the first genome in history in 1995 in the human genome were actually new mathematical algorithms as much as the technology. The technology allowed us to generate the data, but it's the algorithms and the computer that we built that allowed us to actually put it together. In 1999, my team built the third largest computer in the world that now you can buy a card for your desktop computer that's as powerful as one we had in a room this size. So it's changing very rapidly. Is it totally there yet? No, but you know, it's there enough to start this process.
So,
I think there's a lot of progress that's been made on data compression algorithms, and these are, but by changing the kinds of files that you store from a sequencer. Early on when we launched the first sequencers that only did 1 G of output, everyone wanted to look at the raw image data that came off these sequencers. And
we used to print them.
we used to print the images, right. Yeah, and so these files were absolutely gigantic. And today that's technically unfeasible to take the image files off the kinds of sequencers that exist today. We can get down very soon, we think, to the point of where all we store are the differences between the reference and the genome you happen to be sequencing. And if you get to that point, there's some complications around that because of insertions and deletions, but if you can get most of the genome stored that way, now you're starting to deal with files of reasonable size. But I think the key point Craig hit on almost, I think, was the fact that we're going to get very rapidly now to the point where humans aren't going to be able to interpret the data sets that we're creating. You know, he's not going to be having someone sit down or 10 people sit down and say, go look at these 20,000 genomes and all this phenotypic data and tell me what you find. Right? It just isn't going to work that way. It's impossible. And so the machine learning algorithms and the closed loop systems and the artificial intelligence and all these other technologies that are being brought to bear are going to be absolutely necessary to make sense out of the data sets we're creating.
Bob?
For complex diseases for which many genes interact, it's almost certain that it's mathematically chaotic. That is, there's extreme dependence on initial conditions, such as subtle differences in expression level, interactions with the microbiome. 2 questions about that. One is, do we have the mathematics to enable us to model and predict that? And 2, how can we measure, once we've sequenced out someone's genome, subtle differences in expression that may change how the genes interact?
Well, it's what I was referring to earlier of, you know, people that measure these subtle differences in epigenetics act like that's not part of your genetic heritage. It is, right? That came with your genome. It's a matter of learning those rules. You know, right now, we don't even have a way to measure the subtlety. As the data sets get large enough, those associations will fall out.
And
you know?
Do you have the mathematics to do that? Because if it's chaotic, it's not.
No, I think you know, I think the biggest change we'll see in 10 years is we'll be using mathematics that don't exist today. And we're trying to hire a mathematician and an algorithm scientist. That was our key at Celera. That's what made the difference, was our algorithm team, as much as any of the other technology. The sequencing could have been the best in the world, and I think it was at the time. We had the fastest computer in the world. Without the algorithm that Gene Myers and colleagues built to assemble the DNA, it would have been the most historic failure in science.
Okay.
So I love mathematicians. They made it work.
You talked quite a bit about cancer as kind of the frontier.
Can you hear? Can you talk a little bit louder? Is it on? There
Here's
we
the
go.
other end.
Thank you.
I think we used cancer as an example of kind of pushing some of the limits, and you talked about the ALK fusion and discovering drugs, and you talked about circulating tumor DNA as approaches. But what are the conversations that you're having with the regulatory agency about if you diagnose a patient very, very early in the disease, and you think about chemo prevention, chemo intervention, that's really been the dearth of developing drugs. And survival has always been one of the key endpoints in cancer. You're talking about possibly treating patients for years and years to see whether or not they have an outcome. What are the regulatory discussions that are ongoing in thinking about intervening early in the disease outcomes and how you think about treatment paradigms?
I can tackle it a little bit. You know, Bert Vogelstein, who I think is one of the best cancer scientists in the world, he's at Johns Hopkins University, gave an excellent talk here in San Diego just a couple weeks ago, talking about how development of colon cancer is a 30-year process for the first 27 years, it's 100% treatable. If it's detected in that first 27 years, you can totally prevent the disease. If you wait to the last 3 years, you're in totally different territory. So it's a matter of getting smart enough, whether it's circulating DNA, whatever it is. We have this 27-year window that it takes to develop colon cancer. We just have to get a lot better at knowing what to look for in that time period. I think the regulatory agencies are going to depend on what we're trying to generate. Large data sets that are accurate and are meaningful and become predictable. Without that, it's been very slow and cumbersome. We've had inaccurate data. We've had sloppy predictions. The genetics community, if they find a 1% association between a SNP and a disease, publish that in the scientific literature. It should never even be published. So we have a system that requires a much higher standard, and we're already starting to work with the FDA because we want to help them establish standards. Jay's been working with them. Greg's been working with them. We want to establish the standards for the world, and we want it to be a high standard. We had this X Prize that, in fact, I think helped spawn this entire industry. After we sequenced the first draft of the human genome, analysts were saying the sequencing industry was dead and there was no value to it whatsoever. My institute started a $500,000 prize, and that's caused people to turn around. And the early guys at Solexa were, they wanted to get that $500,000 prize, and then with Peter Diamandis and the X Prize, we raised that to a $10 million prize. I think it helped influence this entire industry. We recently canceled the prize because these guys got so big, they could care less about a $10 million prize. I was able to influence Jay's behavior more by placing a big order with him than I ever could with a prize.
Jay? Yeah, we work a lot with the FDA, and I think they're at the point where they realize their paradigm in the regulatory world has to shift. They're just trying to figure out how to get from where they are to where they need to be, and there's a big leap here. I think in the LDT world, what you're going to see over the next few years is that these oncology diagnostic tests are going to get deployed broadly in the LDT world, and I'd be quite surprised if the regulatory agencies clamp down on those tests, because they already are saving lives, directing treatment, and making huge differences. So I think that would be a highly unpopular move for the FDA to take. However, in the IVD space, where today you have to have large-scale clinical trials that are statistically significant, I think particularly in oncology, the paradigm does have to shift, because many times the variants in genes that might particularly cause a disease or cause a drug reaction may be unknown for that particular patient. And Myriad has shown this in spades. They started with a handful of variants in the BRCA genes, and their database is now 18,000 variants. And so you're going into a world where you don't necessarily know what the variants are, and a clinical trial doesn't. It might get you there in a fraction of the population, but it certainly won't get you there for all of it.
Greg, any?
No, I think that's well said. This controversy of an IVD versus LDT, a few years ago, the FDA was going to shut down the LDT world. And I think, as Jay says, genetic sequencing, particularly in cancer, has caused them and requires them to completely reevaluate the whole LDT space.
Okay. Guy.
One of the things, one of you made a comment earlier about knowledge is power, and reacting to Greg's comment that in 10 years, for example, we'll bring something back from the dead in terms of extinction. I personally think that in 10 years, these moral issues will be the key things, because the data sets will get so big that a lot of these issues will be resolved in terms of the functionality and utility of using genomic decision-making in medicine. And I think it will devolve to the individual, the married couple, the unmarried couples, the people who are planning their lives forward. And I think looking to China, the consequences become really, really clear. I think what's going to happen is that the decisions will become staggering and difficult, because we'll have the ability to do just about anything we want to. And this opens up a really a conundrum, a Pandora's box of issues. Could you talk to this? Because I find this much more intriguing, because I think the technology will sort itself out.
Yeah, I mean, I agree it's going to, at some point, it'll hopefully be driven by a personal choice, and every individual will have that choice. I think the ethical dilemmas will be Gattaca-like, at least, if not worse, than what was represented in that movie in terms of the power of what the underlying technology can do. We don't have the answers to this. I think it's a topic for ongoing debate. One of the reasons that we launched our consumer sequencing program 4 years ago at $48,000, and people thought we were crazy. Our investors said, why are you doing that? There's no market. And we said, you're right, there is no market. But the reason we did it was to try to catalyze the discussion around these issues. And I think hopefully in some small way it has. And there are now lots of debates about these ethical questions. And it seems as though it takes society, at least our society, to get very near the point where it's going to be overwhelming for the issues to really get addressed. And I think we have to sort of push it to that limit, and then people will start making the calls and sort of sorting out the legal environment, the ethical environment, the moral questions. And that may depend on your religion, what country you live in, and all of the surrounding factors.
There was a great article in the New York Times years ago when John Roberts was under review to become the Chief Justice of the Supreme Court. And they were asking him questions about his views on abortion and all these present-day issues at the moment. And I thought the New York Times did a great job writing an article about this guy's going to be in his role for 20-plus years because he was under 50 at the time, and we're asking him all the wrong questions. Because in his tenure, he's going to have to decide the rights of a clone. And, they went on and on about some of these very serious potential questions a decade or two from now. And so I would just simply say, you're right, these are the big issues, and it is going to require really great thinking, great frameworks to come up with the right decisions, because just about anything's going to be possible.
Craig?
I mean, it's hard to argue with what either of them has said, and it's the kind of question we've been asked from the start of genomics, and to me, it's more of a question of when versus if, which is what my colleagues are saying. And I hope most of the applications are not for changing relatively trivial traits as to whether you have sprinter genes in your muscles or distant running genes, or we will be able to predict the facial looks, hair color, eye color, all the things that people sort of focus on as the trivial components. It would be sad if this powerful technology was used primarily for those purposes versus trying to eliminate some of these devastating diseases that, sooner or later affect everybody. We all die from something, and trying to understand that, and that's what human longevity is about. Can we get to healthy aging versus just extending life? It's not trying to make better traits, but all biology is based on selection. And pre-implantation diagnostics is just following that selection paradigm. But what you're selecting for become society decisions, cultural decisions, moral decisions. Are you selecting for sex? Are you selecting for, what traits are important to you, and what becomes the standard of the medical community? And these are going to be big debates in society. And it does need regulation at some stage, because I don't think it should just be left up. Everything should be left up to individual decision, or pretty soon, well, it becomes self-limiting. If everybody in China has a male child,
we win.
We win. Unless they're all angry.
Yeah, well,
Okay, we're going to have one more question. Take the lady over here.
Okay, now I'm really going to have it end on something big, because I'm sitting here with incredible people, just intense minds and solutions. So I am from Illumina. I work for Jay. And I went to work there about 6 months ago. I am at risk for Huntington's, and I have not been tested. I hear all of everything that you all are saying, and I've been scared in the closet. I don't want to tell anybody this, right? Insurance, a job, I'm the main breadwinner. I've participated in studies at UCSD. I'm a blind study person. They know I don't, for the last 15 years. So I've been trying to contribute that way. I want to make a difference, and I want you all to tell me, because I hear you say, it's that power, know what you're dealing with. But Huntington's there's not a solution yet. So how do you, Jay knows, we just launched a program at Illumina for the employees to be tested at no charge. At no charge, guys. Seriously, it's phenomenal. And I've been walking around Illumina for my months now, making a difference, being very happy to be employed there. It's a wonderful company. But no one knowing inside I have this personal issue. And when they came out with this program, I went immediately and read everything, and then I thought, but I don't want Illumina to know I'm at risk for Huntington. What if I test positive for the gene? So these are the issues. And I hear you guys and I go, wow, here I am. I want to hear what you have to say to help me understand. Genetic counseling, absolutely. I believe that's huge, because look at, I would need that, and my family too.
Well, I've dealt with this a lot and thought about it a lot for a long period of time. And it used to be relatively simplistic when the Huntington's gene was found. As with all genetic diseases, you talked about breast cancer, all the different alleles. As you know, because I'm sure you've studied Huntington's disease a lot and have dealt with it probably in your family, that there's totally different ages of onset. There's totally different types and degrees of severity, that just knowing you're at increased risk at some stage will be powerful information to have. And there are subcultures where, depending on the size family you're in and whether your family, other members have it, you find out you don't have that risk. People have guilt associated with that. So there's all kinds of cultural things associated whether Jay is going to fire you because you have a Huntington's test. I don't think he will. In fact,
we will. Especially now.
Smart girl.
Yeah. This was one of the biggest concerns early on with the genome, and the NIH didn't want me to disclose my genome on the internet, and we had to go through almost a year and a half of an IRB process where I could make that personal decision of disclosure. And everybody was worried about employers and insurance companies using your genetic information against you. Louise Slaughter sponsored a bill. It took her about 15 years to get it passed. We and I think my colleagues here helped with that. The genetic non-discrimination bill, where not only should he not do it, it would be illegal for him to fire you or take any kind of actions because you have an increased genetic risk for something. And the same is true for denying people insurance, health insurance, based on that. And whether you like Obamacare or not, number one thing it does, it makes it no preconditions. Mm-hmm. Guess what? Everybody in this room, if we sequence your genome, you got preconditions. Yeah. And now it's not an issue, right? So at that level, there's no fundamental issues. Will people sometimes discriminate? Yeah, that's what the legal system is for, to get it so that it doesn't happen. I think the social and society issues around you and your family are ones that nobody can answer for you. I can't answer it for you. We can give you the probability, we can give you the information, but guess what? The more people that contribute their data to databases and to public projects are part of the solution for the next generation, even if it doesn't solve it for you. Everything will become actionable in the future. There's been more research on drugs to treat Huntington's disease since the gene was discovered by Nancy Wexler and her team, what, 20 years ago now? And so, you know, information becomes power at some stage.
You know, first of all, I commend you. You have a lot of courage to ask the question and reveal information about yourself like you have. So I really want to say thank you for being so transparent and forthright. And Jay, honestly, I think it's terrific you're offering that program to your employees. We did the same at Life in terms of the impact program for families in our midst that contracted cancer. And our whole view, and I'm sure it was yours as well now, is if we're going to be a company that is espousing the future of genomics, we have to be the company then that walks the talk. And I can't tell you how many families we were able to touch in terms of where knowledge made a difference in terms of the treatment of that family's cancer, and such appreciation back to the company for pushing the envelope. But I think this dialogue there where Craig was answering your question is just, again, we're on the forefront of so much more of this. And I think if this symposium was held, 3-4 years from now, we will have moved beyond such questions and concerns. And I think that's what is incredibly positive and bright about the future we're moving into. That we will overcome these concerns, that we will push the boundaries of human health for the better. He's going to do it, he's going to do it. And I think San Diego, just to finish that point, is a great place where it's all going to take place. So, I'm very hopeful for you, and again, I'm very hopeful for all of us here about what we're heading into.
So anyway, thank you. Hasn't this been a great panel? Would you please give them a round of applause? Thank you.